Now, as you can see, it is written the internal structure of the anther. So it is today's topic that we would be dealing with the internal structure of the anther and the microsporogenesis. Now, what is microsporogenesis? Microsporogenesis, basically it is a process by which pollens develop within the anther. That means the production of male gametes in the flowering plant. Now here you can see the diagram. Now here this diagram, basically it is a diagram TS. It is a TS of a anther lobe, and it is a dioecious anther lobe. What is dioecious? It is a two-lobe structure. Now you can see the structure. It is bilocular. Bilocular means if we divide it, we find two lobes here and two lobes here. And in a central part, this is TS of an anther, and the central part is known as made up of sterile tissue. The center part is made up of sterile tissue, and here what you see this side is known as the anther wall. Now firstly, we begin with the anther wall. If you see from here, the anther wall, the outer layer of the anther wall is made up of epidermis cells. These are all flat cells, a single layer flat cells of epidermis layer, and this gives the function of protection. What is protection? Protection to the anther lobe from desiccation. What is desiccation? Dehydration. It gives the protection. Now, after the epidermis layer, the inner layer is known as endothelium layer, and this endothelium, it is not possible to draw the structure wholly, but you can have an idea. You see, these are vertical parallel lines I have drawn, so that means this depicts that these are the endothelium layer, where single layer of cells are there, but these cells are radially arranged. And in the middle of these two radial cells, the white space given is filled up of callus thickening. And what is callus thickening? I'll come to the later part. Now after this layer, you can see small triangle like structure. So this is known as the middle layer, and this is made up of loose parenchyma cells that is arranged in 3 to 4 layers. Okay, now after that, the center part of this cell is known as the tapetum, and this tapetum are made up of pyramidal cells, and it has got a very special function. It is here. The pyramidal cells is known as the tapetum, and here before that pyramidal layer is 3 to 4 layers of parenchymaceous cell, and here the tapetum, they are pyramidal cells. And what is the function of the tapetum? They are very special functions. Now what are the special functions? The special function is they form a large polypoid nucleus. This pyramidal cells, they are made up of large polypoid nucleus or multinucleated structure. Now you may say, why multinucleated structure? Actually they have got a very thick cytoplasm, and endomitosis and karyokinesis happens. That means the nucleus is divided without the division of the cytoplasm. That is the basic structure of the tapetum cell. Now why is this tapetum layer has got a tapetum cell has got a very thick cytoplasm? Why? Because this thick cytoplasm is made up of nutritive layers also. So why does this nutritive layer is there? Because it provides nourishment to the growing anther. Okay, now you can see here inside this layer. Okay, inside this layer you find small round small dots over here. Okay, so these are what? These are actually the growing anther cells that would be undergoing microsporogenesis, and here inside that you can call it as a microsporangium. Now what I was telling, that these tapetum has got up from here to here, you see they are 4 layered structure, but at this junction they doesn't have that layer. So this portion is known as Dehiscent zones. So all these 4 layer makes these tetrad means 1, 2, 3 and 4. So they have got a pack of 4 microsporangia in each anther, and these junctions, the cell wall is very thin. You can see why this cell wall is thin. This cell wall, this is the cell wall that is known as, that helps in dehiscent of anther. That means these are the junctions that breaks apart for the release of the anther, and this portion along with all these cells, they makes the surface more rigid. Okay, now you see the small round structure, they are actually the developing pollen grains. And outside structure of each pollen grain, you find a very tough structure that is known as sporopollenin. Now that particular sporopollenin, the outer covering of the pollen, that forms the exine, E-X-I-N-E, exine. Exine of the pollen is supposed to be the toughest substance, and it prevents physical aberration of these pollen grains. Okay, now inside this pollen grains you have another cell that is known as pollen kit, that has basically 2 layers, the glandular and the amoeboid. So they actually secrete certain enzymes. Now one thing I forgot, that is they have, see, tapetum has got, secretes an enzyme that is known as a callose digestive, that is callose, and this callose is actually help in digestion of the wall here when the pollens are released. Okay, so in the next part, we would be dealing with microsporogenesis in detail at the genetic level. This is a gross outside structure. You go through it properly and then we'll come to the next part.